going to tell us something very related, also in a very short talk. So I've got about 13 minutes on the clock here. Just make sure that this is on here. There we go.
Can you guys hear me?
All right, take it away.
Is the microphone good? Okay. Thanks, John. And so yeah, I'm Purushottam. I'm a postdoc at Columbia. And thanks for inviting me at this conference. I used to be a structural biologist, but I've moved to systems biology now. But I've kept some interest in building models from limited data. And so when I was preparing a talk for this conference, I was in two minds whether I should focus on just the molecular dynamics aspect of these kinds of methods. But then I decided to go otherwise, and I'm going to give you just a brief overview of how these inference methods work, and the example that I will use would be from molecular dynamics. So as most of you know, Markov models are ubiquitous in both structural and systems biology. A lot of talks here have been about understanding dynamics amongst metastable states of biomolecules. But we're also interested in Markov models, let's say in biochemical reaction networks, where you're trying to figure out how genes are expressed from the DNA. And more recently in understanding how protein sequences evolve. And in all these cases, the challenge that one faces when you're trying to build a Markov model is that the data is often noisy and incomplete. For example, when it comes to biomolecular dynamics, the information that you would have, let's say, would be the equilibrium populations of these metastable states that one can get, let's say, from a enhanced sampling calculation. And you may have some experimental information about the average dynamics of the system, let's say a folding rate and an unfolding rate. But what you wouldn't know a priori is the whole rate of transition matrices. And similar is true across different examples, where in biochemical reaction networks, you would estimate the distribution over different proteins, but you wouldn't know sort of the dynamics, what's happening inside individual cells. And same is the case for protein sequence evolution, where what you know is a whole bunch of sequences, and you want to build like an evolutionary dynamics models on that. And what I've been interested in is understanding whether we can take this kind of incomplete data and build from scratch a Markov model, or in another set of questions, if you already have a Markov model, this is similar to what Simon was talking about. If you already have a Markov model and if it doesn't agree with your data, what are the ways to update that model using some kind of a framework? In physics and in information theory, we already have such a framework, and it's called the principle of maximum entropy. I'm not going to go over what this means. I'm guessing most of you are somewhat familiar with it. Usually the maximum entropy principle is used on the state space. So let's say in a protein it would be updating the stationary probabilities of individual states. But there's also a dynamical way of working with maximum entropy, and it is to maximize the entropy of the ensemble of paths. So for a cartoon, you can imagine that there is a graph. This could be a graph of metastable states, and imagine a walker that can jump or hop around this graph. And you can consider long trajectories of this hopper, and the entropy over the space of these trajectories would just be given by your standard formula. Now, if the dynamics of this hopper are Markovian, the entropy can in fact be decomposed in terms of the stationary distribution of the hopper and the transition probabilities of the hopper. So pi a here is the stationary distribution of some Markov process, and PAB is the probability of going from state A to state B in one time step. And the idea would be to take this entropy functional, add some constraints, I'll talk about the constraints in a second, and then maximize the entropy to find the transition probabilities. So the constraints could be of various forms depending on the kind of problem that you're interested in. You may know the stationary distribution that you want to enforce on this Markov process, which would be, let's say, for example, a Boltzmann distribution. Now, if you're working with molecular dynamics, you may also want to impose detailed balance, which would make sure that the forward trajectories look similar to the backward trajectories. In case if you're working with a non-thermal system, you may want to just constrain the population averages of some property instead of imposing an entire stationary distribution. And finally, you could choose to impose dynamical constraints, which would be properties that depend on transitions of this Markov process. And the idea is that you would maximize this guy while imposing some of these constraints using the standard method of Lagrange multipliers. And what you get out of this, and we have shown this over a whole bunch of papers, is that you could get perhaps transition rates. If you do it in proper space, you can also get back Smolachowski partial differential equation. So I'm going to give you a very small example of a quick and dirty way, which seems to work, and I was very surprised with this, a quick and dirty way to get to the transition rates of Markov state models just by using an ensemble of equilibrium populations and very short simulations. So let me first tell you sort of the mathematics behind this. So you have your path entropy and you maximize it with respect to many constraints. And let me just walk you through the kind of constraints that are relevant for this kind of problem. So first set of constraints are row normalization. So what you want is that if you start from any state A, at end of one step, the probability of reaching to all other states adds up to 1. So this is the fact that the probability matrix is a row normalized stochastic matrix. You can impose the stationary distribution with respect to a known probability distribution. Then you can impose a path ensemble average, and this would be some kind of a dynamical property of the protein. And finally, you can impose detailed balance. And you can carry out the crank of actually figuring out what are the transition probabilities that are consistent with all these constraints. And you can do this across multiple scenarios. So if you're looking at, let's say, discrete time, discrete space Markov processes, you can derive the transition probabilities in terms of some Lagrange multipliers, and they can be evaluated in a self-consistent manner. In fact, if you're looking at continuous time, these Lagrange multipliers can be evaluated analytically, and you get the transition rates now in terms of the square roots of the probabilities. I think Pratyush Tiwari yesterday talked about this particular formula. And more recently, we have actually shown that if you take appropriate limits of this guy further into continuous space limit, what you get out of it is a Smoluchowski differential equation, which has a position-dependent diffusion coefficient. And in fact, that diffusion coefficient can be analytically derived in terms of the dynamical constraint that you have imposed on this system. So this is sort of how the framework works. We tried to apply this framework to study, to get a very rough idea of how dynamics of small molecules may look like. So we have collaborators in Gerhard Stock and in his lab who gave us this long simulation of this very small helical peptide called AIB, and the idea is that this peptide has a left and a right conformation and basically goes from one conformation to the another. We had access to the whole molecular dynamics simulation, but in order to get to just the basins of attraction or the metastable states, you can disregard the dynamic information and just look at the densities of states in the phase space and figure out what are the approximate metastable states. And you can get from this data the stationary distributions over the metastable states. Then what we did was we ran very, very short simulations, and these were, I think, 200 picosecond simulations from different parts of this phase space. And all we asked was, in those 200 picoseconds, how far did the protein travel in some internal representation? And we got some kind of dynamical average from that data. Now, this did not require a very long simulation. All it required was many, many short simulations. We ran like thousands of, around 1,000 short simulations. Now, what I want is a Markov process which is consistent with this equilibrium distribution and this dynamical ensemble average. And using the cranks that I showed you earlier, we predicted what the Markov process would look like, and this is the result. So on the x-axis here are the rates that we predicted using our Markov process, using our maximum entropy method. These are on the log scale, and on the y-axis, these are the rates that one observes in a full molecular dynamics simulation. And this is obviously not entirely accurate, but given the amount of effort that we put in this calculation, I thought it was fairly remarkable. Then we went on to do this for other proteins, slightly bigger proteins. So we've done this for the Willen headpiece and the BPTI protein. Obviously, since these are bigger proteins, you don't get as good a prediction. But again, given the amount of time we actually saved by not doing these long simulations, it seems like a quick and dirty way to actually get a very rough idea of how the dynamics will look like. And I would be very interested in seeing whether this kind of approach can go further. Now let me just end this brief talk by giving a cautionary tale, and it actually helps. Simon's talk actually helps a lot here. So, for example, in his work, what they did was they had an observable which was a stationary state observable. Now there are two ways of imposing a stationary state observable onto a dynamical process. You can first take the stationary state observable, change your stationary distribution and impose that entire distribution onto the Markov process. or you can directly impose the stationary state observable onto the Markov process. And these are very two different situations. And we've tried to do that. So now you can also impose, let's say, so for example, if you have a polymer and you have some information about the size of the polymer in a solution, you can impose that size onto the stationary distribution and then project that stationary distribution on the Markov process, or directly impose the size itself on the Markov process. And when you carry out that particular way, so now you can determine both the transition probabilities as well as the stationary distribution that is consistent with this information. You get a very different problem, and it turns out to be an eigenvector problem where the solution of the stationary distribution looks like the Schrodinger's equation. And this is kind of scary because this is not going to be very useful for thermal systems. So to give you a very brief idea, so let's say if I construct a Markov process on a 2D grid where a particle is supposed to hop between nearest neighbors. Now, and if I want to construct a Markov process that is just consistent with this piece of information, one would expect that the stationary distribution of this Markov process is basically flat on the entire 2D grid with some corrections on the boundaries. But when I actually carry out the crank, you see that the stationary distribution has a nice peak in the middle and is actually in fact identical to the ground state of the Schrodinger equation on a 2D grid. So it would be important to understand how one actually incorporates these constraints using these kinds of inference methods, because they could have very non-trivial effects on the final outcome of the inference method. And finally, let me thank all the people who were involved. So I've started working with Ken because of our common interest in Markov processes, and he's been great help in doing a lot of these projects. I'm not very good at molecular simulations, so whenever we need some simulations, we ask Professor Gerhard Stock to give us the data, and these are the papers if you guys are interested. And sometimes the money came from NIH. Thank you, all of you.
The correlation of the time scales.
Yep.
So we see, the time scales that are probably most important are probably the slowest rates.
Yeah.
And there we see a systematic issue, right, that the Markov models are overestimated. Are you doing any plans for that? Or?
So the way we do it is that we have used a very specific dynamical variable, and all I'm calculating is just in the dihedral angle space how much is the protein moving. But obviously, as you impose more and more dynamical constraints, this is supposed to get more accurate. So I'm basically searching for, but those dynamical constraints are going to be more system dependent, I guess. So what we have been systematically doing now is searching for constraints that are appropriate for those two systems, which would allow, so where we would constrain the space of Markov models, and they're forced to recapitulate some of the data that the MD simulation produces, so to see whether we get back these slow processes or not.
Yes. To add to the last question, so for AIB and HG35, can you say something? So these are the microscopic kind of rates. Can you say something about the long, like the folding time?
Oh, so I actually haven't. I'm actually in the process. So this is not published. So I'm actually in the process of doing exactly that.
Alex.
Could you clarify something with the dynamical constraints? It's on the last slide. So when you're calculating that delta,
Yeah, that one.
Oh, maybe it was the slide after. It was where you're right there.
Oh, okay.
So this ensemble average delta depends on the weights of the states.
Yeah.
So are you kind of solving for the weights self-consistently with kind of
No. So the idea, So the idea here is that you could probably get the weights using just pre-energy calculations, which can be highly parallelizable and cheaper than getting the full dynamics. So in these things, I'm assuming that the weights are available to you prior, and yeah. So the task is to get to the rates that are consistent with those weights.
Great first.
So I was curious, maybe just my limited ability to read equations quickly, but like if you have the folding time for a villain, like can you incorporate that as a constraint, or do you need to be able to formulate things to like have specific start and end points for your paths?
So
that would be like an eigenvalue of it.
So clearly you can. The one thing one has to worry about is that some of the experimental constraints may not express themselves as convex quantities in the probabilities. Right? So in the standard formulation of maximum entropy, we require that the constraints are convex, and then you get this nice exponential form. So if a folding time is between one state and another state, that's going to be a constraint that is that's nonlinear in the probabilities. You can enforce that using brute force and some kind of maximum likelihood way, but you may not get an analytical form out of it.
Avi?
Oh, okay. Is there any way to incorporate like error into this?
Yes. So actually, I didn't get time to do that. So let's see. So in that paper, so it's basically it's the same thing. Isn't errors? So there could be two types of errors: one in the measurement and also in determining the transition probabilities. And so I worked it out, and basically it looks exactly like what Simon did, with slight modifications because we also have dynamical constraints now. But
yes, kind of interesting, right? Because there's these two types of errors that you have, right? You have like, especially if you're doing like experiment or simulation, like one of them is that different dependencies, right?
Yeah.
One is like an actual measurement error, and like a simulation error that decreases the more you simulate, right?
Yeah. So the simulation error.
Interesting interactions.
Yeah. So
two types.
yeah, the way I've done is that the simulation error comes in the form of prior, that and the experimental error comes as a likelihood, and then you can evaluate the posterior. So yeah, it's in that paper. So.
Thank you.
All right, let's thank Purushottam.